Hi everyone, Dr. Hinkie here for our final section of Chapter 1, our Introduction to Microbiology. We're getting a big broad overview of everything that we'll be covering. So right now, getting familiar with the terms, so just generally recognizing these are the concepts we'll be exploring in more depth is what we're going for. So if as you read these or hear about the organisms, say, "Oh, I don't know anything about these." That's right, because we're at the beginning of class. So that should be how you feel. But we're building some foundational knowledge that we'll expand on as we go. So let's take a look at our PowerPoints for this section. And there we are. So section 1.3 just covers the types of microorganisms that are studied in microbiology. Our objectives for this section: by the end of this, you should be able to list the types of microorganisms and acellular infectious agents and their characteristics. You should be able to describe the similarities and differences between archaea and bacteria. You should give examples of different types of cellular and viral microorganisms and infectious agents, and provide an overview of the different areas in the study of microbiology. So first, if we look at microbes, what we're talking about when we talk about microbiology and microbes are things that are not visible with the naked eye. And this image shows us this scale that we're talking about. We don't become visible to the naked eye until we're at about 100 micrometers. So frog eggs, you can see those in the pond. They're bigger, they're visible, an average about 1 millimeter. Human egg, that's the largest cell in the human body. Very tiny, but at about the limits of visibility as well as pollen. A typical plant and animal cell, there's a lot of variation here as we can see, will range between 10 micrometers. Some are smaller, like red blood cells, and some will be larger, but between 10 micrometers and 100. We look at this scale along the bottom of this image. Each unit is a 10-fold increase. So this isn't just twice as big, 3 times, 4 times. This is 10 times larger, 100 times larger than this first atom, 1,000, and so on. 10,000, we go up by 10 each unit. So a red blood cell is about 1/10 the size of other animal cells. The mitochondria, an organelle within eukaryotic cells like ours that's responsible for producing energy, is 10, 100, 2 units smaller, about 1/100th the size of the entire cell. Mitochondria are similar in size to many bacteria, and we'll talk about why that is in our next chapter. Then, as we can see, there's a range of viruses. We have a very small poliovirus, a flu virus, smallpox virus, but even the smallpox, one of our larger viruses, is smaller than a bacteria, almost half as large. And then, of course, if we think about this scale as we go down, our viruses are complex infectious organisms. We'll learn all about those, but they're made of a nucleic acid surrounded by a protein. So they're made of multiple different types of molecules organized in a specific way. That means they contain proteins, that protein code. So proteins must be smaller. Proteins are a macromolecule, a big molecule, but they're still smaller. They're the building blocks of larger things. So they are one of the building blocks that make up viruses. So obviously proteins, the building blocks, have to be smaller than the entire thing. Lipids, for example, our plasma membrane that surrounds all cells is made of a phospholipid bilayer. They must be significantly smaller if it takes millions of them organized in an envelope to surround a cell. So when we look at that, molecules must be smaller than the things they make up. And atoms, which make up molecules, must be smaller than those molecules. So this just sort of makes sense as we go down here. So our living things, when we talk about microbes, we're looking at microscopic organisms, things that are not visible to the naked eye. And really, we're looking down at this end. We will look at some eukaryotic cells. There are some some eukaryotic cells that we'll study in microbiology, those that are microscopic, or at least at one stage of their life cycle. So the specific organisms we cover, primarily bacteria. We spent a lot of time with bacteria. We think about bacteria, again, smaller than our own eukaryotic animal cells, about the size of one of the organelles in our cell, the mitochondria, the powerhouse of our cell. Most bacteria are harmless to humans, and in fact, the majority of bacteria are helpful. They recycle nutrients, they remove waste and toxic materials from ecosystems, and they serve as the primary producers for many food webs. Some bacteria are pathogens. A pathogen is an organism that causes disease. That will be the focus because we are a clinical microbiology class. Most of you are preparing for healthcare fields. So while we focus on pathogens, just know that some are pathogens, but the majority are not. The majority provide helpful services and are essential for our existence and survival. So bacteria have a wide range of metabolic capabilities. It can grow in a variety of environments. We already talked about the fact that bacteria are ubiquitous. We find them everywhere. The archaea, this is our other domains. We have our domain bacteria, our domain archaea, and then our domain eukaryotes. The eukarya, nucleated cells, organisms with nucleated cells. The archaea do not have a nucleus. They are similar to bacteria in size, but we've discovered that they have distinct molecular differences in their membrane makeup, in their cell wall makeup, pseudo-peptidoglycan, as opposed to the cell wall of bacteria, which is peptidoglycan. There's sort of this transition species between the bacteria and the eukaryotes. They have some of their components that are more similar to eukaryotes than they are to bacteria. These are often found in extreme environments, so we sometimes hear them referred to as extremophiles. So very cold, very hot, very basic, very acidic, high pressure, high salinity, environments that other living things would have difficulty living in. You find archaea have adapted to those and survive and thrive in those. There are no known pathogens in the archaea, but as we learn more and more about those, we are finding that they aren't all extremophiles. Well, many of them can be and are extremophiles. They're the group we're most likely to find in these extreme environments. They also live in some not very extreme environments, including on and in our bodies. But none of these are known pathogens, so we won't be spending a whole lot of time on the archaea. So if we look at our bacteria, the way we differentiate our bacteria often is based on morphology. This is our first differentiation. Remember, we talked about taxonomy, and we start with domains, and then we classify to more and more similar and more similar until we get down to genus and species. Some of the very first classifications that we do to separate into smaller and smaller and smaller units in our taxonomy of bacteria. First is shape, and that semi-rigid structure, that peptidoglycan, gives us a rigid external support structure. So the bacteria have distinct shapes because of that cell wall. Some common morphologies that we'll see a lot in lab and we'll talk about frequently are spherical. Coccus, cocci, plural. Bacillus, which are rod-shaped, or bacilli in the plural. Vibrio are comma-shaped, so comma, slightly curved bacillus. We can have cocobacillus, cocobacilli, which are not quite as oblong, sort of this cross spherical, but a little bit more oblong, slight modification. Kind of a combination of our Coccus and Bacillus bacteria, so cocobacilli. And then we have spirillum, which are spiral shaped and flexible, and spirochetes, which are very helical, rigid, like a corkscrew structure. That's our general broad overview of bacteria and archaea. Our prokaryotes, pro before karyote, the nut or the seed, that nucleus. So we also have some eukaryotes that we study in microbiology. Our eukaryotic microorganisms, many of them fall into the eukaryotic kingdom Protista. Remember, we have the kingdoms Animalia, Plantae, Fungi, and Protista in our eukaryotes. The protists, this is a kingdom that's not based on evolutionary history and relationships, but based on we don't know what else to do with you. So we're going to put you in this single-celled kingdom for now. Now that we have this great molecular technology and can look at these organisms on the DNA level, on the molecular level, looking at their biomolecular makeup, their DNA and RNA, we're able to figure out what those evolutionary histories are. And so in taxonomy right now, we're in the process of dividing these into supergroups based on an evolutionary history. So this is really an influx kingdom at the moment. But right now, we subgroup the protists basically by how they get their nutrients and their energy. So within the protists, we have some single-celled organisms that are plant-like. They're photoautotrophs, capable of photosynthesis. We include the algae in here. Most algae are single-celled phytoplankton, aqueous, floating in the oceans, but even the macroalgae fall into the protista group. They're the one exception to that single-celled rule, because algae don't have the same complex structures that plants do. Then we have the fungus-like protists. They are saprobes. They neither eat nor photosynthesize, but they absorb nutrients from the environment around them. They release exoenzymes, enzymes outside of their body that break down dead and decaying organic matter, and then they absorb the nutrients. So they acquire their nutrients and energy in a fungus-like manner. They're saprobes, so some things like water molds and slime molds. And then we have the animal-like protists. We refer to those as protozoans. They're animal-like. They're chemoheterotrophs. That means they eat other living things. And then we have some that are both photosynthetic and heterotrophs. They have the ability, if there's no food available, they can photosynthesize. They have chloroplasts, that photosynthetic organ that plants have. And if they're stark or if there's food around and it's easier, they'll eat other organisms. Euglena are an example of that for anyone who's taken 101 or 102. So our protozoans, the animal-like group, are primarily the ones we'll focus on. We won't look at algae at all. Protozoans are very diverse. Some are free-living. Others are obligate internal parasites. They must live inside a host. And these are the ones that can be pathogenic typically. Often we differentiate the protozoans by how they move. Whether that's their pseudopods, fake feet, by pushing out their cytoplasm, making cytoplasmic extensions and then pulling themselves forward with those, or through flagella, a whip-like tail. Cilia, short oar-like extensions, hair-like extensions, and some are non-motile. They don't move in their adult phase. So we'll look at protists, typically single-celled protozoans. Fungi is another group. We think about mushrooms on our pizza. Well, they're fungus, but they're not microscopic. We do have some that are multicellular, not microscopic, bread mold. Lots of fungus that we can see as a mold, especially those are the multicellular form. But we also have some single-celled varieties, yeast, single-celled fungus. Many fungus are dimorphic, meaning they can either be a single-celled yeast fungus or they can be multicellular, made of hyphae, long threads, multicellular threads, like we see with bread mold. The molds, so they can be yeasts or molds, either one. And often the environment, the conditions in the environment, the temperature, the pH, the moisture will determine which form they take. Fungus are not photosynthetic, as we talked about with the protists that are fungus-like. They acquire their nutrients by decomposing dead or decaying organic matter in the environment around them externally and then absorbing it. And then another group of eukaryotic microorganisms we'll look at are the helminths, worms. These are multicellular. They can be very large, 3-meter long tapeworms. We study these in microbiology and they're considered microorganisms because many of the disease-causing helminths have microscopic eggs and larvae, and that is primarily when they are transmitted is through the release of eggs and larvae, often in water, and then that's how they get into a new host and grow into that larger macroscopic worm that we think of when we think of worms. We also have an acellular group of microorganisms that we study. Acellular, that prefix A means not cellular. We'll look at some other types as well, but viruses are the primary one we'll talk about. We'll also talk about prions. But viruses are obligate intracellular parasites. Oftentimes you'll see them referred to as obligate intracellular infectious chemical compounds because they're not living. They have to use their host cell to multiply. It's that word obligate, means we must be inside a host cell. Outside of the host cell, viruses are inert. We say they are acellular. They're not composed of cells, so they're not considered living. So in order to be considered cellular, we have to have cellular level of organization, meaning we have to have cell membrane surrounding cytoplasm, aqueous solution that has dissolved substances that a cell requires, the resources it needs, the nutrients it needs, all the building block compounds it needs. And within that cytoplasm, we must have DNA. Got to have the instructions for how we will metabolize or function. We have to have ribosomes. Ribosomes are organelles where we make proteins. Since all our chemical reactions, all of our metabolism requires proteins, enzymes for us to function. Without those ribosomes and ability to build our own proteins, we don't meet many of the requirements that it takes to be considered living. So all living things have organization, and they have the cellular level of organization at least. Viruses do not have that. Metabolism is the sum total of chemical reactions in a cell. So must have the ability to take small molecules and build large ones, and the ability to break down large molecules to release energy to do work. Viruses have no metabolism. They're unable to carry out chemical reactions on their own. They must use the host cell's machinery to do that. Living things have to be able to reproduce. Viruses can only reproduce when they're inside a host and use the host cell's machinery. They do have adaptations because adaptations just refer to the accumulation of mutations that allow an organism to survive. So because when they replicate inside the host cell using the host cell machinery, mistakes can happen. We can have mutations. They do have those. And another criteria for living things is they have to be able to respond to stimuli. This is a big debate among biologists and virologists uh, because the only interactions that viruses have with their environment are chemical reactions. So is it a legitimate response to stimuli, or is it just a chemical reaction that would have happened anyway? It's not an actual motivated response to stimuli based on senses. So there's some debate on whether or not that chemical reaction is a response. So these are the 6 criteria for something to be classified as living, and you have to meet all 6 of those. Viruses do not, and so they are not classified as living, only as obligate infectious particles, obligate intracellular infectious particles. But viruses will spend quite a bit of time on viruses. So, our areas of study. When we talk about microbiology, it's a broad term and there are lots of subdisciplines. So all those different types of microbes can be their own area of study. So we can study bacteriology. We study bacteria, structures, functions, metabolism of bacteria. Mycology, mycologists study fungus. Protozoologists study protists, right, are protozoans. Parasitologists often study both protozoans, so parasitic protozoans, as well as helminths. Virology is the study of viruses. And immunology is included in this because that's a study of an infectious agent, the interactions between an infectious agent and a host immune system. So those are the main broad areas, but microbiology can also be divided into subdisciplines based on a habitat or environment of the microbe or its specific function. So we can have marine or aquatic microbiology, in my background, marine microbiology. Environmental microbiology, a little broader, not just in aqueous environments. We can have agricultural microbiology, looking specifically at microbe interactions with agriculture. Geomicrobiology, we study the biogeochemical cycles that bacteria and archaea and other microbes are intricately involved in. Aeromicrobiology can study the bacteria that are present in the air. We can focus on specific areas, just like with agricultural microbiology, you can focus on veterinary microbiology or astromicrobiology, looking at microbes that may exist outside of our atmosphere on other planets in space. Those are all different areas or subsections within microbiology that people can focus in. It's one of my favorite quotes. If you remember in our previous unit, we talked about Carl Woese and Fox. They used ribosomal RNA to come up with the most recent and most accurate phylogenetic tree that we have, helping to divide out the archaea and the bacteria, seeing those differences. And so Carl Woese, it's a great quote about why are we studying microbiology? Why should we care? And hopefully this will pique your interest a little bit so that you are taking this class not just to check this box off, not just because you have to do it to get into the nursing or some other allied health field, but because this is what you will be doing in those health fields. This is what you will be doing every day. So it's intricately involved. It's important that you know this, but also, why should we care? We can't see these things. Well, let's do a thought experiment and take these things away. Let's take all the bacteria in the world away. Do you know what's going to happen? All the life that we can see with our naked eyes, all that life is going to disappear in short order. The microbial world is the basis upon which our whole ecosphere rests. And without it, there's no multicellular life. And the humbling thought is, if you do the converse and take away all the animals and plants, you'll see big adjustments in the bacterial world, but it will still be there, growing happily along and evolving. We need them, but they don't need us. It is a humbling thought, isn't it? and also shows the importance of understanding microbes and their function. All right, so let's go ahead and check our learning from this short section. Match each description with the appropriate microbiology subdiscipline. So A, the study of how antibodies are released in response to a pathogen. Which of those subdisciplines studies antibodies, response of our immune system, the presence of a pathogen? Just said immunology. The study of fungi, including their genetic and biochemical properties, taxonomy, and their ability to cause illness or disease. So fungi, the study of fungus. Mycology. But the study of the morphology, ecology, genetics, and biochemistry of bacteria. Just going to add ology, bacteriology, and the branch of biology or medicine concerned with the study of parasitic organisms. Parasitology. Good. All right. A blank is a disease-causing organism. So now everybody should know the correct answer to that is not a bacteria. Not all bacteria cause disease. Germs is a generic word that really means nothing. We want to be more specific. What's a disease-causing organism? Correct term is a pathogen. Which of the following is not considered a microorganism? Mosquitoes, bacterium, protozoans, or fungus? Well, mosquitoes can be small, but they're not microorganisms. And fungus can be large, but not all fungus are. So we do study those. So mosquito is not considered a microorganism. About this one, which of the following is true of the microbial world? All microbes are harmful. Humans could survive without microbes. Many microbes are helpful. Or most microbes are pathogenic. Hopefully everyone now understands that many microbes are helpful. The study of viruses is called virology. Okay, just going to add ology. And which of the following indicates how bacteria and viruses compare in size? Bacteria are indistinguishable from viruses. Bacteria are much larger than viruses. Bacteria are much smaller than viruses, or bacteria are similar in size to viruses. Hopefully everybody remembers that chart that we started out with that shows bacteria are much larger than viruses, 10 to 1,000 times larger. So quick review, our learning objectives. But at this point, everyone should be able to list the types of microorganisms and acellular infectious agents and their characteristics, the things we'll be studying. Describe the similarities and differences between archaea and bacteria. Generally, so pseudopeptidoglycan in archaea versus peptidoglycan in bacteria. No archaea cause disease. Some bacteria can be pathogens. And then there's some other distinct molecular differences in their cell membrane, their ribosomes, and their function. You should all be able to give examples of different types of cellular and viral microorganisms and infectious agents, and provide an overview of the different areas in the study of microbiology. This third one and the first one are kind of saying the same thing, but hopefully you are all ready to do that. And now you are hopefully very excited for what's to come as we dive further into all of these things we briefly touched on in chapter 1. Okay, we'll see you in chapter 3.